Hi everyone, in this video we will revise questions from vitamin B6 and biotin. First question is, what is provitamin? Give example. Provitamins are precursors of active form of vitamin. They are present in food substance and when they are ingested in the body with the help of enzymatic action, they are converted to vitamin. Example for provitamins are beta carotene. Beta carotene is a provitamin of vitamin A and this is present in plant sources, and when ingested in the intestinal mucosal cell, an enzyme beta carotene dioxygenase convert it into retinol. And 7-dehydrocholesterol, it is the precursor of vitamin D. So these are the examples for provitamin. One is beta carotene, which is a provitamin form of vitamin A, and 7-dehydrocholesterol is the precursor of vitamin D. Second question is, mention examples for antivitamins. So antivitamins are chemical compounds which will inhibit or oppose the vitamin activity and produce vitamin deficiency. So examples for antivitamins are dicumarol and warfarin. They are antivitamins of vitamin K. So this warfarin and dicumarol, they will competitively inhibit the gamma carboxylation system due to its structural similarity with vitamin K. Another example for antivitamin is avidin. So avidin, it is a protein present in egg white and it has a great affinity to biotin. It is one molecule of avidin, it can combine with 4 molecules of biotin. Other examples are oxythiamine and methotrexate. Next question is antihemorrhagic vitamin. Vitamin K is known as the antihemorrhagic vitamin because it is necessary for the blood coagulation. And factors like factor 2, 7 and 9, they are dependent on, and factor 10, they are dependent on vitamin K. Coenzyme form and functions of pyridoxine. The active coenzyme form of pyridoxine is pyridoxal phosphate, that is PLP. And PLP as coenzyme is required in many reactions like transamination reaction, decarboxylation, sulfur-containing amino acid metabolism, heme synthesis, production of niacin and glycogenolysis. So transamination reactions are catalyzed by a group of enzymes known as aminotransferases. And this aminotransferase, they require pyridoxal phosphate as the coenzyme. All decarboxylation reactions of amino acid metabolism require pyridoxal phosphate as the coenzyme. And these are the example for decarboxylation reaction. Glutamic acid, it is decarboxylated to GABA. GABA, it is an inhibitory neurotransmitter. And other example for decarboxylation reactions are tyrosine to tyramine, histidine to histamine. All these decarboxylation reactions require pyridoxal phosphate. ALA synthase is the rate limiting enzyme in heme synthesis, and this enzyme, it is a PLP dependent enzyme. So B6 deficiency, it accounts for anemia. Then PLP has got a role in the metabolism of sulfur containing amino acid, that is the enzyme cystathionine synthase and cystathionase, both are PLP dependent enzymes. Then it is required for the production of niacin. The enzyme kineurinase, it is a PLP dependent enzyme. Then glycogen phosphorylase enzyme of glycogenolysis, that is also PLP dependent enzyme. Now coming to the deficiency manifestation, the vitamin B6 deficiency can be neurological manifestation, dermatological manifestation and hematological manifestation. Neurological manifestation, here the PLP dependent enzyme, they function poor. Serotonin, epinephrine, noradrenaline, GABA, these are neurotransmitters and they are not produced properly. So there will be neurological symptoms. And this PLP, it is also involved in the synthesis of sphingolipids. So the deficiency of B6, it will lead to the demyelination of nerve and consequent peripheral neuritis. Dermatological manifestation. Deficiency of B6, it will affect the tryptophan metabolism because niacin, it is produced from tryptophan. And B6 deficiency, it will lead to niacin deficiency and this is manifested as pellagra. Then hematological manifestation. There will be hypochromic microcytic anemia because the enzyme PLP is essential for the synthesis of heme, that is the enzyme ALA synthase, which is the key enzyme for heme synthesis, is PLP dependent. Next question is coenzyme functions of biotin. Biotin, it is required for carboxylation reaction, that is carbon dioxide fixation reaction. And this carbon dioxide fixation, it occurs in two steps. First one is binding of carbon dioxide to biotin to form carboxybiotin. Then this carbon dioxide, it is transferred to the substrate. So the examples for biotin dependent carboxylation are, one is propionyl coenzyme A carboxylase, which converts propionyl coenzyme A to methyl malonyl coenzyme A. Another example is acetyl CoA carboxylase. So this acetyl CoA carboxylase, it will add carbon dioxide to acetyl CoA to form malonyl coenzyme A. And this is the rate limiting step in the fatty acid synthesis. Third example is pyruvate carboxylase. Pyruvate carboxylase, it converts pyruvate into oxaloacetate.